Assalamu alaikum. This is a tutor from Ilm Online Academy, and I'll be teaching you biology, IGCSE Biology paper, June 2022. So let's get started. The first question says some students were studying the activity of yeast and they made a fact file and organism. They found that organism was yeast. Scientific name was Saccharomyces cerevisiae, and with that structure is single cellular structure. Main cell structures क्या हैं? Those are, it has got cell membrane, cell wall, vacuole, nucleus, mitochondria, and rough endoplasmic reticulum as well. Now, main energy source for yeast is sugar. Name the kingdom in which yeast is classified. So they are asking you about which kingdom yeast is classified. So you would have studied it in the first chapter. Yeast is classified in the kingdom fungi, and I hope that you're familiar with the kingdoms that are that you I mean the kingdoms that you have actually studied. So mainly those are kingdom animalia or animals, plants, plants, fungi, in which yeast falls, and then prokaryote and protoctist. Protoctist or protist, two names for this kingdom. Okay, so okay, protoctista भी कहते हैं इसको. Okay, if we talk about kingdom animal or kingdom animalia, kingdom animal में क्या होता है कि the organisms would not have cell wall in their structure. Now right here you have seen that yeast has got cell wall in its structure, but normally if we talk about kingdom animal, तो क्या होगा? It won't really have cell wall in there. The animals won't have cell wall in their cellular structure. But when we talk about fungi, तो fungi में cell wall होती है, but the cell wall is different from that of the cell wall that is present in plant cells. Fungi will have cell wall made up of something other than cellulose. Plants में जो cell wall होती है, that is actually made up of cellulose. And the cell wall is that simply means cell wall here is made up of cellulose in plants. And then if we talk about the difference between the fungal cell and the plant cell, to remember that plants are autotrophs. Mostly plants are autotrophs, so they would have chloroplasts and the chlorophyll as well in their structure, but clearly yeast does not have that. So it is from kingdom fungi, and yeast is not a prokaryote. Prokaryotes are the organisms that do not have defined nucleus. Their genetic material would be floating in the cytoplasm. Then we have got protoctist. So protoctist में कौन आ जाते हैं? Protoctist में होते हैं euglena and paramecium and chlorella, these organisms. State the process that occurs in mitochondria to provide energy for yeast cells. Achha. So remember that when we talk about yeast cell, so yeast cell mainly does anaerobic respiration normally. ठीक है, that simply means yeast जो है, it can actually respire without oxygen. But the main thing is that yeast can actually respire with or without oxygen. That simply means it can carry out anaerobic as well as aerobic respiration. Now, anaerobic respiration of yeast takes place in the absence of oxygen, and it actually takes place in the cytoplasm of the yeast. ठीक है? And we also call it fermentation. But when we are talking about the process that occurs in mitochondria to provide energy for yeast cells, so for sure, if it is providing energy, it is respiration, because in respiration is a process that provides energy, right? But in mitochondria, aerobic respiration takes place. So in mitochondria, you should know that mitochondria is the powerhouse of cell, and in mitochondria, aerobic respiration of yeast takes place, and a lot of energy is produced as a result of it. But remember, in anaerobic respiration, what happens is that yeast will use glucose and produce carbon dioxide and alcohol, but not a lot of energy. But it won't produce a lot of energy. But the aerobic respiration taking place in mitochondria would help out in producing a lot of energy. So the answer would be aerobic respiration. Okay, that's the process that takes place there now. Now, next question is, yeast cells make enzyme sucrase. Sucrase catalyzes the breakdown of sucrose to glucose and fructose, and enzymes are made of protein. Explain the shape of sucrase molecule is relative to its function. Now remember that the shape of an enzyme, it is an enzyme, right? It is an enzyme which actually breaks down sucrose, catalyzes the breakdown of sucrose. So if we talk about enzyme, the enzyme is really specific for the shape of the substrate, right? It is really specific for the substrate that fits into its active site, or it is really specific for the substrate with which it is going to whose reaction it is going to catalyze. Like sucrase जो है, that specifically acts on sucrose, right? Okay, enzyme, the active site of this enzyme actually is designed for sucrose to come and bind to it, and that way catalysis will take place and sucrase will actually catalyze the breakdown of this molecule, right? So enzymes, you can write it as enzymes are specific type of substrate that the shape of the so shape of the active site of sucrose is complementary to the substrate. Sorry, sucrase is complementary to the substrate sucrose to bind to it, and that way catalysis takes place. That's that. Then the students made an extract of sucrase from yeast cells. They investigated the activity of the sucrase extract at different pH values. They determined the rate of reaction at each pH. They then calculated the rate of each reaction as a percentage of the fastest reaction to give the percentage activity of sucrase. So the results of an investigation have been shown in front of you, right? Let's see the graph. So percentage activity of sucrase at pH 2, you can see it's at this mark. Then at pH 3, it increases to being like above 60, and then above 70 at pH 4. Then at pH 5, it's like 80, and then at pH 6, it is. Now this peak is actually telling you at pH 6, the activity of the enzyme is maximum. Okay, and then when the pH increases even more, the activity decreases, right? So the pH actually increases when the activity of the sucrase of enzyme increases as the pH goes from pH 2 to 6, and it is maximum at pH 6, and then it decreases from 6 onwards to 12, and at 12 the activity of the enzyme is minimum. All right, so you're just going to it's a 4-mark question. You're going 6-mark question. You're going to try to write as you need to describe and explain the fact of activity on the effect of pH on the activity of sucrase. So try to write like about a few points, six points. All right. So for the description, try writing like 4 points and then two for the explanation. That's what the marking scheme mainly says about how to score well in this. So what can we write? We can say that the enzyme is active between pH 2 to pH 12. Okay? It is active between pH 2 and pH 12, and the activity increases. And you know that the enzyme has the maximum activity at pH 6. It has the least activity or minimum activity at pH 12. These are 3 points. You are not really going to write it in the point form, like you're going to make a paragraph out of it. So A of sucrase, sorry, sucrase is active. According to the graph, sucrase is active from between pH 2 to pH 12. At pH 6, the activity of the enzyme is maximum, and at pH 12, its activity is minimum, so it works best at low pH rather than a high pH. And you can even say that the activity is more activity is in acidic conditions and less activity in alkaline condition. That would be a similar thing. And उसके अलावा other than that you can say so this happens because the change in pH enzymes, because enzymes are sensitive to a change in temperature or pH, and they work best at their optimum temperature and pH. So this is because enzymes work their optimum in optimum environments and are sensitive to change in pH and temperature. So the change in pH change in pH actually affects the shape of the enzyme. Remember that when we talk about the activity of enzyme getting less or enzyme getting inactive, that simply means that the shape of enzyme is actually getting changed, or the change in pH or temperature is actually affecting the shape of the active site of that enzyme, and that means the shape of the enzyme has been partially denatured, and for that reason what would happen is that enzyme substrate complex is not going to form. And if enzyme substrate complex is not going to form, then that enzyme substrate reaction would not take place, and you will not get your relevant product, or enzyme would not really uh, react in the way it needed to. So a change in pH affects the shape of the active site of sucrase, and at extreme pH it gets partially denatured, and enzyme substrate complex cannot be formed. So eventually the activity decreases. Okay. Then mammals have a double circulation, and this is the diagram of a section through the heart of a mammal, and the arrows show the direction of blood flow through the heart and blood vessels. Okay. And you can see in this diagram, you can see there are a lot of parts which we need to label. So if we talk about this part, it is aorta. See, this is aorta. If we talk about A, so it is pulmonary artery. And then B, pulmonary vein. Okay, then X is semilunar wall. Vena cava. Okay. State the name of the chamber of heart with the thickest wall. So that is left ventricle, and the reason why left ventricle has the thickest wall is that left ventricle, it is actually the main powerhouse of the heart now, and it's responsible for most of the pumping that takes place, pumping with the blood, pumping with the blood towards the other tissues of the heart, towards the other body parts and the other tissues of the heart. So the stronger, thicker muscle helps to actually produce higher force to move blood towards the system. So it helps that way. And D is actually a vein, and D जो है that is vena cava. ठीक है, and describe the structure. Its name is vena cava. We named it just now when we were seeing the diagram. Then we need to write the description about its structure. So vena cava जो होती है, it is actually, it has a thin wall, as well as it consists of it has got a wide lumen. With that, it consists of like elastic connective tissue as well, and it is very large vein. Remember that it is very large vein. And इसका वैसे function, it's just that it brings like deoxygenated blood, blood and in order to heart, in order to get oxygen. किस तरह से कि deoxygenated blood from heart is going to go to the lungs, oxygen, it will become oxygenated and it will come back to the heart, and from heart then it will be transported to the other tissues or the parts of the body where it is required, right? And then the cycle actually continues this vena. If you have studied about double circulation, so that's that. Okay, so um, it has a thin wall, wide lumen, and consists of elastic tissue. All right. Then identify the structure labeled X and state its role in the heart. Okay, so the structure that is labeled X is semilunar wall, and If we are to talk about its function, let's just write firstly down. It is semilunar valve, and what it does is that it prevents backflow of blood. It prevents backflow of blood from the arteries to ventricles during the ventricular diastole. So it actually prevents backflow of blood from arteries to the ventricle. Okay. Okay, that was the one. During ventricular diastole. Then next question is the diagram that shows the double circulation of a mammal, and the arrows indicate the movement of oxygen and carbon dioxide in and out of the blood. Or okay, yes, it is double circulation. And remember that there are two sides. We call it double circulation because you know that there are two sides or two parts of the heart, right side and left side, right? And those two sides of the heart are actually separated through a wall that is septum, right? So before now, let's see the first question. It's shade the blood vessel that transports blood with the highest oxygen concentration. So that is pulmonary vein. You just need to shade that. And pulmonary vein is this one. Okay, this is pulmonary vein. So just shade this one. Okay. Then describe the evidence shown in figure 2.2 that mammal has a double circulatory system. So mammals have a double circulatory system and we need to see the figure. So remember that what we are going to write is just that your heart has got two sides. Okay, you can see that here. Heart has got two sides, left side and right side. And these two sides are actually separated through a wall that is septum. And then what happens is that कि जो right side of the heart होती है वहाँ पे the deoxygenated blood actually goes from the heart to the lungs, and when it goes to the lungs, ठीक है, यहाँ पे lungs की तरफ आया and it will get deoxygenated blood था, it will get oxygenated, and when it gets oxygenated, the oxygenated blood is going to move is going to go back to the heart, and oxygenated blood is going to move back to the heart, and from the heart then that oxygenated blood is going to get pumped to the other parts of the body, because now it's oxygenated, it has got oxygen, the oxygen would be oxygenated blood is going to go to the other parts of the body, or it's going to reach to the cells where respiration needs to take place, or the parts of the body where energy is required. Okay? Respiration is the process through which body gets energy, in which glucose and oxygen combine to give carbon dioxide, water and energy. So this oxygenated blood is going to reach there, and and फिर वहाँ पे cells में क्या होगा कि cells में that oxygenated blood is going to get give its oxygen to the cells, and then it's going to become deoxygenated again, or when it becomes deoxygenated again, so what happens is that it's going to return back to the from body to the heart again, and then it would be sent to the lungs back for oxygenation, right? So this is going to continue. Achha, this was all about like the double circulation, and I hope this would help you out in answering this question as well. Okay, so what are we going to write? We are going to write heart has two parts. sorry, two sides, left and right, which are separated by septum. And right now, I showed you that what happens is that blood passes through the heart twice in order to complete one cycle, right? Okay, it started off from here, and then this blood went to the lungs, and then it got oxygenated, then it returns back to the heart, and then it goes back to the blood, and then it returns back to the heart. So जो blood होता है, it has to go back to the heart like twice. It has to, in order to complete one circuit of the body. There are two circuits, pulmonary circuit and systemic circuit. I'll just try and show you a diagram as well for better explanation. So let's see. Okay. Okay, we'll see that later if we are left with that. Okay. So, in fact, this diagram even works here. Okay, let's answer the question. Heart has two sides, left and right, which is separated by the septum. And there are two circuits, pulmonary and systemic circuits. Blood must flow through the heart twice in one circuit. Twice in one circuit. The two circuits are pulmonary. But 2 points are enough, but just writing it that just in case. Pulmonary and systemic circuit. Now, these two circuits, like one of these, as I have mentioned even before, so one of these actually carries, like it's from heart to lungs, blood from heart to lungs, and then heart to rest of the body is the other circuit. So that's that. Then explain the advantage of a double circulation. Now, double circulation, so what happens is that oxygenated and deoxygenated blood are kept separated. Oxygenated and deoxygenated blood are kept separated. Separated. And how are they separated? They're kept separated by septum. And that actually ensures double circulation actually ensures efficient supply, of oxygen to the body. And if efficient amount of oxygen is supplied now, so it actually results to ensure that there is efficient rate of metabolism or fine rate of respiration taking place, though helps in maintaining the rate of respiration. And helps in rate of respiration. It also ensures efficient supply of nutrients. So you can write it as efficient supply of oxygen and nutrients in order to make it smaller. Supply of nutrients. And efficient supply of nutrients and blood to the body. And there are a lot more things. It would allow more time for gaseous exchange, and it's going to prevent like damage to the capillaries that are present in the lungs, and lower pressures in the pulmonary artery and circuit mainly. So, and it also allows the quite many things, right? So yeah, 4 points, 4 to 5 points are going to be enough. Then the question number 3 says the pancreas is an organ that has roles in the digestive and hormonal systems of animals, and this figure is actually showing some of the parts of alimentary canal and some of the associated organs. So J is liver, K is L is duodenum, okay, and K is your gallbladder. Okay. Gallbladder, and it is duodenum. Duodenum actually then extends to small intestine, so you can write it either way. Then hormones secreted by pancreas. So hormones are insulin and glucagon. They actually balance the levels of sugar levels in your body, insulin and glucagon. And enzymes secreted are carbohydrates, trypsin, and other than that, lipase. So carbohydrates को हम, if we want to name it, like trypsin is a protease ना mainly. So carbohydrates mainly amylase, trypsin protease, and then lipase. So amylase will break down carbohydrates like starch, complex carbohydrate like starch, and trypsin actually is a protease, proteins ki breakdown, and lipase would work on lipids. Okay, so we'll continue from here in the next part. That would be it for this video, and Allah Hafiz.